Both clonal expansion and clonal differentiation contribute to immunological memory in B cells. Immunological memory in B cells can be examined by isolating B cells from immunized mice and re-stimulating them with antigen in the presence of armed helper T cells specific for the same antigen. The response of these primed B cells can be compared with the primary B cell response seen on isolating B cells from unimmunized mice and stimulating them with antigen in the same way. By these means, it is possible to show that antigen-specific memory B cells differ both quantitatively and qualitatively from naive B cells. B cells that can respond to antigen increase in frequency after priming by about 10 to 100-fold and produce antibody of higher average affinity than unprimed B lymphocytes. The affinity of that antibody continues to increase during the ongoing secondary and subsequent antibody responses. The secondary antibody response is characterized in its first few days by the production of small amounts of immunoglobulin M antibody and larger amounts of immunoglobulin G antibody with some immunoglobulin A and immunoglobulin E. These antibodies are produced by memory B cells that have already switched from immunoglobulin M to these more mature isotypes and express immunoglobulin G, immunoglobulin A, or immunoglobulin E on their surface, as well as a somewhat higher level of MHC class II molecules than is characteristic of naive B cells. Increased affinity for antigen and increased levels of MHC class II facilitate antigen uptake and presentation and allow memory B cells to initiate their critical interactions with armed helper T cells at lower doses of antigen. Unlike memory T cells, which can traffic to tissues owing to changes in cell surface molecules that affect migration and homing, it is thought that memory B cells continue to recirculate through the same secondary lymphoid compartments that contain naive B cells, principally the follicles of the spleen, lymph node, and Peyer's patches. Some memory B cells can also be found in marginal zones, though it is not clear whether these represent a distinct subset of memory B cells. The distinction between primary and secondary antibody responses is most clearly seen in those cases where the primary response is dominated by antibodies that are closely related and show few, if any, somatic hypermutations. This occurs in inbred mouse strains in response to certain haptens that are recognized by a limited set of naive B cells. The antibodies produced are encoded by the same VH and VL genes in all animals of the strain, suggesting that these variable regions have been selected during evolution for recognition of determination on pathogens that happen to cross-react with some haptens. And that's both clonal expansion and clonal differentiation contribute to immunological memory in B cells. Have a great day.